Hello everyone, welcome to 10-Minute Physiology. In today's video, I'm going to give you a short explanation as to how the GPCR pathways work. In other words, the G protein coupled receptors. So with that, let's begin. So I'd like to start off by first giving you a short explanation as to how these G proteins actually look. So this right here is the plasma membrane, and it separates two fluid compartments. The first is the intracellular fluid, and the second one is the extracellular fluid. Now, inside this plasma membrane, we have the G protein coupled receptor. So this is the receptor part of the G protein coupled receptor. So the receptor of the G protein coupled receptor is basically a really big protein that has 7 transmembrane domains. So these rectangles are the parts of the receptor that span the entire length of the membrane. Now, all of these transmembrane domains are connected to each other by these chains that we see here, or loops. And the receptor also has 2 tails. The first tail is the N-terminus tail, which is in the extracellular fluid, and the second tail is the C-terminus tail, which is in the cytoplasmic fluid. Now, the G protein coupled receptor also has G proteins coupled to it. So this is the receptor part. And in addition to that, we have the G proteins themselves. So the G protein is going to be made up of 3 parts. It's a trimeric protein. So the first part is the alpha subunit. And the alpha subunit is what we're going to be mainly focusing on in this video, because the alpha subunit is going to be the main part of the protein that will do all of the effects. The second subunit is going to be the beta gamma subunit. Now, the beta gamma subunit in this case it has basically 2 roles. So the first role is that it acts as an anchor for the alpha subunit, and the second role is that it actually can have different physiological effects. So it can actually go to different channels and different places and exert its effects. We're not really going to be talking about the beta gamma subunit in this video. We're going to save that for when we get to specific organ systems. Now, the alpha subunit in its inactivated state is going to be bound to GDP. And when a ligand comes in and binds to the receptor, it'll cause a conformational change to go throughout the whole receptor into the proteins, and it will basically cause a conformational change in the alpha subunit, which will decrease the affinity of the alpha subunit for GDP, causing it to dissociate, and in its place will come GTP. So GTP binds to the alpha subunit, and this is when the protein is activated. So in other words, the G protein is now activated, and now it's going to start to do its effects. So once GTP binds, the beta gamma subunit will dissociate, and the alpha subunit will go off and do its effects. So now we're going to talk about the effects of the alpha subunit, and the effects of the alpha subunit depend upon what type of alpha subunit it is. So the first one we're going to be looking at is the G-alpha stimulatory pathway. So what is that? So this right here is the alpha subunit, but in this case it's the alpha stimulatory subunit. So the alpha stimulatory subunit, which is now activated because it's bound to GTP, is going to target a specific enzyme called adenylate cyclase. So adenylate cyclase is going to be responsible for converting ATP into something called cyclic AMP. So when the alpha stimulatory protein goes and binds to the adenylate cyclase, it stimulates it, which therefore causes the adenylate cyclase to convert ATP into cyclic AMP. Now, inside the cytosol, we have another protein called pKa, or protein kinase A. It has 4 domains, 2 catalytic domains, so these are the domains that actually produce effects, and 2 regulatory domains that are bound to it and keep the catalytic domains together. So these regulatory domains, when they're bound to the protein, basically prevent protein kinase A from exerting its effects. So cyclic AMP will bind to each domain, which will cause the regulatory domains to fall off. And when the regulatory domains fall off, the catalytic domains are free to go and do their thing, which will therefore lead to the effects. So they're going to phosphorylate different proteins, leading to different effects. So the second pathway is sort of the antagonist of the previous pathway. This is the GI pathway, and otherwise known as the alpha inhibitory pathway. So here we have the alpha subunit, but this time it is an inhibitory alpha subunit. And what's going to happen is that you have adenylate cyclase. And remember that adenylate cyclase converts ATP to cyclic AMP. So the alpha inhibitory subunit will bind to the adenylate cyclase and inhibit it. This will cause a decrease in the conversion of ATP into cyclic AMP, which will decrease the level of cyclic AMP, which will therefore lead to a decreased pKa activation, which will then lead to certain effects. So the GI pathway is basically the antagonist to the alpha stimulatory pathway. And then the last pathway that we're going to talk about is the GQ pathway, or the alpha Q pathway. Now this is the most complicated out of the three, and we're going to go through this a little bit more slowly. So in this case you have alpha Q, and it's activated because it has GTP, and in this case it's going to go to a specific enzyme called phospholipase C. So phospholipase C takes a molecule that is present inside the plasma membrane and converts it to two different second messengers. So when alpha Q came here and it bound to phospholipase C, it activates it, which therefore causes phospholipase C to catalyze a specific reaction. And this reaction is the conversion of PIP2 into IP3 and diacylglycerol, or DAG. So basically what PLC is doing, or phospholipase C is doing, is it's converting PIP2, which is found inside the plasma membrane, and converting it into inositol triphosphate, or IP3, and diacylglycerol, or DAG. Now, each of these second messengers has different effects, and we're going to go through them individually. So let's look at IP3 first. So we have our IP3, and the IP3 is actually going to go to a specific cellular structure, the endoplasmic reticulum. Now, the endoplasmic reticulum has a specific receptor called the IP3 receptor. And inside the endoplasmic reticulum, you have a ton of calcium. So the concentration of calcium inside the endoplasmic reticulum is way greater than the concentration of calcium inside the cytosol. So this is symbolized by having a much bigger calcium concentration here. So what happens is when IP3 binds to the receptor, it opens it, which then allows the calcium inside the endoplasmic reticulum to flow down its concentration gradient into the cytosol. Now, the calcium then is a second messenger, and it's basically going to exert different effects. So as the calcium concentration increases inside the cytosol, the calcium is going to bind to a specific protein called calmodulin. Now, 4 calciums are going to bind to calmodulin, and the calmodulin will then exert its effects by going to and activating another protein called calcium calmodulin kinase. It's a kinase protein which adds phosphates to other proteins. And then this calcium calmodulin kinase can then lead to different effects. The second messenger is diacylglycerol. Remember, that was produced by phospholipase C. And diacylglycerol is going to work in conjunction with calcium to activate a specific protein called protein kinase C. So protein kinase C is water-soluble protein, so it's in the cytosol. And so what happens is that calcium and diacylglycerol will bind to the R domain, and it will localize this enzyme to the plasma membrane. And when this enzyme is activated by diacylglycerol and calcium, it can then lead to specific effects. So how is the signal terminated? Well, we're only going to look at this in a simplistic point of view because we'll go through how signals are terminated as we go through specific organ systems. But in general, remember that the alpha subunit, when it is activated, is bound to GTP. Now, the alpha subunit has hydrolase activity. So in other words, it can use water in order to cleave certain bonds. And the alpha subunit has hydrolase activity that is timed. So after a certain amount of time, the hydrolase activity will be activated, which will therefore cause GTP to be cleaved into GDP. and a phosphate. So once you have GDP bound to the alpha subunit, the signal is going to be terminated. Now, in addition to the hydrolase activity, you also have other proteins inside the cytosol that can actually quicken up the hydrolase activity. So once GDP replaces GTP, this is when the alpha subunit is deactivated. So when GDP is there, the alpha subunit goes back to its original place, and then the beta gamma subunit follows, and you can restart the process again. So there are other ways in which the signal can be terminated. This includes the fact that receptors can be sequestered into vesicles. The receptors can be inactivated. The receptor synthesis can be decreased. You can also have different signaling proteins that are downstream from the activated receptor be inactivated. Inhibitory proteins that inhibit the receptor or signaling proteins can be activated. The ligand can be broken down. Second messengers can be broken down. For the context of this video, we're just going to keep it to what these pathways are, because we'll get into how these receptors and all these signaling pathways are regulated as we go through the organ systems. So this is a summary of everything we talked about in this video. So we saw that GPCRs are receptors with 7 transmembrane domains. The N-terminus is in the extracellular fluid, and the C-terminus, which is the cytoplasmic tail, is in the cytoplasm. We saw that there are 3 different G-alpha subunits. We talked about the alpha stimulatory, alpha Q, and alpha I. We also talked about how the beta gamma subunit serves as both an anchor, and it has other physiological effects that we'll talk about later. And then lastly, we talked about how the G alpha subunit has hydrolase activity that hydrolyzes GTP into GDP, terminating the signal. So I hope that this video helped you understand how these receptors work and what their pathways are, and I hope to see you next time. Good luck studying. Thanks for watching.